Okay. Good afternoon. Welcome to 137th episode of Creative Online Weekend Talks. I am Supriya Naika, host for the session. Creative. We are a community driven by a shared passion for creativity and a curiosity for acquiring new knowledge. Creative embodies our belief that to be truly active, we must cultivate creativity in all aspects of our lives. Tagline Knowledge Square. Sharing of knowledge is not just beneficial but essential for intellectual growth. Through the exchange of idea and experience, we aim to foster constructive thinking across a various domain, transcending traditional boundaries. Today's topic of creative talk, ocean color and bio-optical complexity. To present this, we have Dr. Arjun Adhikari, sir, with us today. As we explore this subject, we embark on a journey to understand the interplay between oceanic hues and the complex biological process that underpin them. So, further delay, we start the session. Do not forget, you can catch the live stream of our program on our YouTube channel, Creative GBD. I call Divya, creative volunteer, to introduce today's speaker. Over to Divya.
Thank you, ma'am. Good afternoon, one and all present here. I'm the volunteer of Creative. I'm here to introduce to you a remarkable individual whose contribution to marine science have left an indelible mark on our understanding of our oceans, Dr. Arjun Adhikari. Dr. Arjun Adhikari, with a PhD in marine science from Goa University, stands as a beacon of excellence in the field of estuarine and coastal optical and physical oceanography. Dr. Adhikari's journey in ocean color research has been nothing short of extraordinary. His groundbreaking work as a chief scientist in the Eastern Arabian Sea in 2017 and in Baja, California, US Mexico in 2013 earned him the prestigious River Plate Scholar 23 award from the International Ocean Color Coordinating Group. Throughout his illustrious career, Dr. Adhikari has held esteemed professional appointments, including roles as a guest scientist and visiting scientist at the Leibniz Institute for Baltic Sea Research in Warnemünde, Germany. His dedication to advancing our understanding of marine ecosystem is reflected in his numerous research papers, which delve into topics such as bio-optical constituents, radiant heating rates in coastal waters, and aerosol boundary layer dynamics. In 2020, Dr. Adhikari completed his PhD, culminating in a dissertation titled Coupling of Coastal Hydrodynamics and Bio-Optical Color Components of the Tropical Estuarine Ecosystem. His tireless pursuit of knowledge and his unwavering commitment to the preservation of our ocean serve as an inspiration to us all. Please join me in welcoming the distinguished Dr. Arjun Adhikari. Thank you, sir, for giving me this. Thank you, ma'am, for giving me this opportunity. Thank you. I'm welcoming all the participants and welcoming you, sir, for this session. Thank you, ma'am.
Thank you, Divya. Sir, over to you.
Thank you. I'll share my slides. We'll begin. Are the slides visible? Am I audible?
No, sir. It's just showing started screening.
Okay, so it is loading.
Yes, sir. Yes, sir. Visible. Yeah. In slide mode.
Yes.
Okay.
So Good afternoon, everybody. I thank the organizers at Creative for giving me this opportunity to talk about ocean color. As the title says, this talk will be on the fundamentals of what constitutes an ocean color, what information is derived from ocean color, and how is it useful. As in this snapshot of a satellite pass over Africa on your screens, it travels over the Strait of Gibraltar, which appears to be blue, over to the Sahara Desert, which appears to be yellowish, beige color, and then over to the rainforest, that look green, and then again over the ocean. So, with this being said, and with this snapshot, we'll focus on the fundamentals of different colors that we see of the ocean. As we already know, Earth is primarily called a blue planet or a blue marble, as shown in this photograph clicked by the astronauts of Apollo 17 in 1972. It shows the heterogeneity of color distribution of the Earth. Different colors originate from different sources. From this photograph, we can tell that the productive land is green. However, our observations also show that plants can be of different shades of green. So the question begins, how can these shades of green can be segregated over a larger area? Doing this gives us valuable information that can be useful in a variety of ways. One way would be to go to the designated area in a forest and log all the plant information, but that might take some time. Another way would be that we take a snapshot from satellite and segregate different colors, meaning the wavelengths of the visible spectrum, use a general understanding of that designated area of forest, and then develop a technique that can be applied over the entire forest using the fundamental laws of physics and mathematics. An example I show you of the world ocean, where the entire ocean may seem to be blue, but may still have different colors that could be green, reddish, or yellowish, or even sometimes brown. So, in general, Earth appears to be blue, as in blue marble, because it is two-thirds water. The ocean strongly couples with the atmosphere, which is also responsible for the variations in the ocean color that we see in satellite images or even visually when we go out on the cruises. It acts as a biogeochemical buffer, and then the phytoplankton photosynthesis, which is primary productivity that is estimated to be roughly equal to that of the terrestrial ecosystems. So we see that how is it important for the climate, and then what role the ocean color could play when we try to understand through satellite imagery. Having said that, the phenomena of the climate change also alters the ocean color. So recent reports have been that at least 50 to 60% of the ocean, because of the climate change, are becoming more green. How could that be? So for that reason, ocean preservation is a part of the 2030 Agenda for Sustainable Development adopted by the UN. Ocean color was also included as an essential climate variable by the Global Climate Observing System, where chlorophyll A concentration and water leaving gradients, they are the products under the scientific area of biosphere under the subdomain of biogeochemical. So the ocean color is just not the color of the ocean, but has impacts. So it has so we develop an understanding that shows the impact and then moves us to the climate action and then also affects the life on land. To show the functionality of ocean color as a variable, how it works in climate change or climate analysis, I'll show you an intersection of three common variables, that is the ocean color itself, and the sea surface temperature, and the sea surface height. So sea surface temperature is a key parameter in the global climate system, as we all know. SST is measured to understand how the ocean is interacting with the Earth's atmosphere. In the same way, it causes changes in the ocean color itself. And the sea surface height is the height of the sea surface above a reference ellipsoid. So that can be done directly through satellite altimetry. So in order to understand that, we have to look at the intersections of these different variables. So if we look at the intersection of the ocean color with SST, we will learn about the coral bleaching, harmful algal blooms, melting ice caps, decline in private productivity, bloom dynamics for the estuarine coastal areas, and finally and eventually at the human health. If we look at the intersection of ocean color with sea surface height, we learn about the internal waves, transport or flux of matter, pollutants, transport of blooms, and underwater volcanic eruptions, just for say. And if we look at the intersection of the sea surface temperature and sea surface height, we start to look at the dynamics, different dynamical features of the ocean and different anomalies, which actually helps in conditions of storm surges. However, if we look at the intersection of all three, that is in the middle, in purple, then we get into the domain of the essential climate variables, where we can get information of a uh, variety of different processes. And then it is also useful for the climate model assimilation. So it helps with the resource management services like the disaster management, gives us information about the ENSO, that is El Nino, Southern Ocean oscillations, and the climate anomalies. And then the assimilation of this data in the climate models improves the forecasting predictability. So what we do is we utilize the information that is stored in the visible spectrum of light to understand the variations of the ocean color. So as we'll see in the following slides, the different substances interact differently with the visible spectrum in the water. And because of this interaction, different wavelengths of this visible spectrum, which are from blue to the red, they are attenuated or they are diffused differently in the water column. So on your right-hand side, the exponential decay of the visible spectrum shows that most of the light gets diffused or attenuated very close to the water surface. So actually, the vertical profile of this attenuation as a function varies depending on the matter present in water and is almost observed to be exponential in open ocean clear waters. So when the light it enters the water column, the incident light is either absorbed or scattered, which means that the matter present in the water is either absorbing type or scattering type, or it may be both absorbing or scattering type simultaneously. So in a water from different regions of the world ocean, be it the open ocean, coastal estuarine, we know that the medium is always the seawater molecules. And then we have particulates, which are phytoplankton, and non-algal particulates, or if we combine them, that is called total suspended matter. It is also a particulate. And then we have a dissolved matter. Dissolved matter is the colored part of the dissolved organic matter. So all these combined and then collectively called as optically active constituents or optically active substances or bio-optical constituents. They're all likewise. So the variability of these substances in a water column that may result in some specific type of color. And then it is biogeochemically relevant. So for example, if we have more phytoplankton, that may appear to be green. But in cases, for example, of red tides, that is done by the plankton itself, it may appear to be more reddish. So both where green is more towards the productivity of the water column, the red is more towards the harmful effects. And then again, the non-algal particulates that may appear to be more reddish or brownish in color. And then we have the dissolved organic matter, which may appear to be dull yellow or yellowish in color. And then the color will depend on the concentration of these constituents in the water column. And sometimes it may vary depending on the transport. So if you have more transport of, let's say, non-algal particulates for a specific period, that time the water will appear to be more reddish brown. And then in a different process, it brings more dissolved matter. That will, the color change will be to yellowish. So that is all observed in dynamic environments. So now with the understanding of the visible spectrum that is either being absorbed or scattered, and then we'll restrict the wavelengths from 400 to 700 nanometers. On your left-hand side, the spectral absorption of the optically active constituents that are the chlorophyll, which is equivalent to phytoplankton, the CDOM, dissolved organic matter, DSM, the total suspended matter or the particulates, and the seawater molecule. As you can see that except for the seawater molecules, all constituents are absorbing the short wavelengths strongly in the visible spectrum. And then chlorophyll A has a secondary absorption peak in the red wavelengths around 663 nanometers. And then in the case of DSM and CDOM, that is exponentially decreasing at the longer wavelengths. Now let's take an example to understand this absorption pattern. And let's take an example of chlorophyll A. So as you see that the spectral absorption decreases in the midsection of the visible spectrum, that is between 500 and 600 nanometers. So what this means is that that amount of light is not being absorbed by the phytoplankton or not absorbed by the chlorophyll A, but then it is available for either scattering or it may be absorbed by either the TSM or the CDOM. So that will again depend on how relevant the concentrations are for CDOM and TSM. And a seawater molecule remains constant for the absorption. So it doesn't absorb the shorter wavelengths, but strongly absorbs the longer wavelengths. So collectively, this is called as absorption. And then on your right-hand side, you'll see the scattering part, which is quite similar to the absorption. And then collectively, absorption and scattering, they're called as the inherent optical properties. So these are the optical and scattering properties that are inherent, fundamental to these different optically active constituents. Now, in the second group of optical observations is the apparent optical properties. So here, once the so if we revisit the example from the previous slide, all the interactions between the matter and light, the reflected light is leaving the water surface. So it is an integral of all the optical interactions that has happened in the water, at the surface and in the water column. So straight away, the reflected light or the reflectance at any depth will be the ratio of what is upwelled to the downwelling irradiance. So it will show you how much total interaction was in the water column. So that we symbolize with R, and then the EU would be the spectral upwelling, and ED will be the spectral downwelling. And this happens at all the angles in case of a reflectance. So in a condition, if the water constituents stay constant, so it means the absorption and scattering of the seawater molecule is same, but we also make the absorption and scattering of the other optically active constituents constant. Then the ED, the downwelling, changes due to cloud cover, sun location, and or any other environmental conditions. Then EU changes proportionately. So which means that this ratio will always tell you the state of the optical interaction. And then this will also mean that the exponential decay of the visible spectrum in water will also change with respect to the downwelling, that is total downwelling and surface reaching. Therefore, the inherent optical properties and the apparent optical properties are valuable descriptors of the water body itself. So one of the most common and most used apparent optical property is the remote sensing reflectance. And this is something that I think most of the people have heard of. So that is a ratio of the water leaving radiance and the downwelling radiance. The difference with reflectance is that the reflectance happens at all angles, while the remote sensing reflectance is observed over a small solid angle, reaching the sensors and satellite in orbit. So, and then because of the interactions that are happening in the water, the spectral shape of the remote sensing reflectance changes. And then depending on the shape, the color of the water or the ocean color is determined. And that itself is the ocean color. The definition of the ocean color is the spectral shape of the remote sensing reflector is the ocean color. Now, how to measure the inherent optical properties and apparent optical properties? The conventional way is to do a spectrophotometry analysis. We go out in the field, that is, if we go out to the coast, rivers, or any water body, open oceans, and then we collect samples through a skin sampler or a rosette, and then we treat it and then filter it and store it, bring it to the lab and do a spectrophotometry analysis that gives us the spectral absorption of the individual optically active constituents. So now the methodology to do this for each of the optically active constituents is different. So there are certain standard protocols that has to be followed in order to get to the absorption spectra. The other part of the inherent optical property is the scattering coefficient, which is inferred by integrating the volume scattering function and is measured through specific sensors that are scatterometers or imaging spheres. And there are also many commercial sensors available, like EcoBB and VSF series from the wet labs. For the apparent optical properties, an in situ, in water radiometer is often used to generate the spectral shape of the remote sensing reflectance. So, just you see in the image, that is a radiometer assembly that measures the total downwelling irradiance, upwelling radiances, and the water leaving. Plus, if you can have more sensors, we can also measure or estimate the photosynthetically active radiation, and the backscattering can also be included in it. And temperature, salinity, tilt sensors, pressure, everything can be assembled in the same sensor. So that data can be collectively used using this instrument to generate remote sensing reflectance that is defined with the depth from the surface up to certain depth, and then the diffuse attenuation coefficient as well. So we can see what is coming out of the water column as a result of the total optical interactions, and then how the light is being decayed in the water column. So as to know the specifics, so we can say that this optically active constituent is resulting in this sort of shape or this amount of decay in the water column. And then there are remote sensing satellites. So in this table, you'll see the number of sensors and their platforms that are in orbit right now. So from India, we had Ocean 1 and we had OCM 1, OCM 2. In the past, now we have OCM 3 in orbit. That was done in 2022, I think. And then these are the water quality variables that can be defined from each and every sensor. For example, we have chlorophyll, TSM, CDOM, that is the dissolved matter, KD, that is the attenuation of light at the surface, and then the turbidity indices. So all these satellites are valuable depending on where they pass in a region of our interest. It gives you the revisit frequency and the spectral bands, and these are all very helpful in determining or understanding the ocean color variations. So these sensors, they measure the remote sensing reflectance at certain wavebands in the visible spectrum of light, centered at wavelengths of interest. For example, 412, 443, we already know is the primary peak of chlorophyll A. 490 nm, that is where we're getting the maximum intensity of solar radiation. 555, which is the green band, known to understand the scattering by chlorophyll A, and so on. So depending on the purpose of quantifying or what optically active constituent that we want to quantify, synoptically, different wavelengths can be used in different forms to formulate the algorithms or bio-optical models. So as a result, we have a scenario where we can relate to the ocean color in terms of human health. However, the spectral shapes of remote sensing reflectance, they are not that straightforward. We see wide variations. However, there are certain things that remain constant. So we'll focus on that as well. So in this figure, I'm showing you the different shapes of remote sensing reflectance in the visible spectrum. These are all measured in situ in the coastal waters of Goa through a radiometer. And then these are hyperspectral in nature, means that we have measurements at every nanometer of the visible spectrum. Now, so before we'll get into the remote sensing reflectance variability, we'll get a little bit of understanding about the case 1 and case 2 waters classification. So to simplify things, Morrow and Freer in 1977, they looked at different color variations based on the remote sensing reflectance and then the attenuation lengths as well. So in the open ocean case, they saw that the remote sensing reflectance is majorly governed by the concentration of phytoplankton and the products that are derived from phytoplankton and the seawater molecules. But we have to keep one thing in mind, that the phytoplankton concentrations in the open oceans are itself low. And then the derived products concentration are negligible. So the remote sensing reflectance can be said to vary with the concentration of phytoplankton itself. So it becomes a linear case. So that's why we call it a case one water body. However, as we get to the coastal areas, estuarine waters, things start to become a little more complex. Here we have phytoplankton, we have seawater molecules, and then we have the products that are derived from phytoplankton itself. But we have more sources of the products that are the dissolved organic matter, and then the sediments or the non-algal particulates. So because of these different sources, the optically active constituents like CDOM and non-algal particulates, they are being added to the ecosystem, and they cause more complex scenarios in the variability of remote sensing reflectance. So, for example, coastal waters, they may appear to be green because of higher loads of phytoplankton and primary productivity, as we all know. And then the inland waters may be more brown or yellowish because of the CDOM. So this is a basic classification that is being used by the international community in defining the ocean color as case 1 and case 2. But in case 2 itself, there are many more water types, depending on the different scenarios of the concentrations of the optically active constituents. So one thing I should note here is we can say for simplicity that case 1 is a linear case and case 2 is a non-linear case. So we revisit the figure from the couple of previous slides and showing the different spectral variability. So in figure, the panel A, you see that is Pagodastian blue. And then the panel B is the coastal turbid, and the open ocean is blue. So we, in all this remote sensing spectra, you'll see that the peak of the remote sensing reflectance is in the blue region. So that's why the ocean appears to be blue, because that is the color that is being reflected most. In the turbid waters, you'll see in the images that within the same water body, it can be either brownish, yellowish, or green. So you get a multi-facets in the spectral shapes of remote sensing reflectance. And trichodesmin blue, you have a blue color as well, and the green that is reflecting. So to simplify this, then we have to test the relationship between the inherent optical properties and the apparent optical properties, which is the absorption and the scattering and the remote sensing reflection. And then this approach is called as the forward modeling approach, as first defined by Preston Dorper and Gordon in many, many papers. So, and then in the previous slide, I have shown scattering as B, and then here it is BB. So BB relates to the backscattering. So technically, scattering is a sum of forward scattering and backscattering. So here the backscattering, we say that the backscattering is representative of scattering because it happens more. So if we were to write this in simplified form to understand and apply it to the real life conditions, the simplified forward modeling approach, it approximates RRS as a function of the spectral inherent optical properties. It includes a provision as well to explicitly define a relationship between the inherent optical properties and the remote sensing reflectors. So we'll take a case of open ocean and a coastal turbid. So in open ocean, we now know that it only has seawater molecules and phytoplankton and the products derived from the phytoplankton. So we look to the additive form of the total absorption and total backscattering. The total absorption becomes the absorption by the water molecules plus the sum of all the phytoplankton species, and then the products of that is CDOM and NAP that is derived from the phytoplankton itself. So that's why that is in light blue to make it distinct. And the same case is with the backscattering. And now you'll also observe that the CDOM doesn't backscatter; it only absorbs. So that makes things a little more complex. So the property is the property of the CDOM to only absorb. So now as we come to the coastal turbid water, so here we have the seawater molecules, we have phytoplankton, we have products derived from phytoplankton, and then we have additional sources of CDOM and non-algal particulates or the photosuspended matter. So here we don't, so we get it straightforward. And then, for example, A becomes water molecules plus different kinds of phytoplankton, and then different sources of CDOM that has different spectral shapes, and then the different non-algal particulates that are prevailing in the ecosystem. And the same thing happens with the backscattering. So over and all, this is called the additive nature of the inherent optical properties, meaning that they can all be summed up to evaluate the total absorption and the total backscattering that is coming or that is occurring in a water column. So the inherent optical property of the seawater molecules remains constant, which means that we only have to take care of the remaining three. And then it is safe to say that in the coastal turbid waters, the remote sensitive reflectance spectra doesn't co-vary with the concentrations of the CDOM NAP because of the additional sources. So it becomes an integral. The signal becomes an integral of all the absorption and backscattering by all the optically active constituents. So this gives rise to the bio-optical complexity. So these are called as case 2 waters or the optically complex waters or the bio-optically complex waters. These are all synonyms. So it simply means that describing a relationship between the remote sensing reflectance and the inherent optical properties is much more complex than the open ocean cases, where only chlorophyll is partaking in causing the variations in the remote sensing reflectance. So for the application-wise, one of the fundamental objectives in using these spectral shapes of different spectral shapes or remote sensing reflectance is that you want to derive the concentrations of phytoplankton, CDOM, or the non-algal particulates in these environments. Why is that? Because phytoplankton, you can say that chlorophyll A is index of phytoplankton concentrations, and it is biogeochemically relevant in terms of primary productivity. CDOM is the optical part of the dissolved organic matter and can be taken up as a pollutant or because it competes for light, sometimes causing a hindrance for primary production as well. And then the non-living particulates that are sediments and anything, detritus, anything that is non-living and is in particulate form. So these are all different biogeochemical indicators. Once we run into the models where everything is like there are different equations to measure each one of them. So, and then all these biogeochemical indicators or the biooptically active constituents, these relate to turbidity, ecosystem health and services. They act as source or sink of CO2. They are exchanged or they are transported. There's a flux between the land and coastal seas. They affect fisheries and primary productivity, and they affect humans, as in life on land as well. So now we go back to the same figure that we visited in the beginning, where we are trying to resolve the different shapes of remote sensing reflectance to giving us the concentrations or the levels of this. However, as we see from in this figure that the absorption spectra, it doesn't co-vary. Means that they all share the same wavelengths or wavelengths of the visible spectrum. So here the fundamental objective is to derive the concentrations, which is severely challenged because of this nature. That's why it is termed as bio-optical complexity. So we have to use the same ratio, that is RRS, this, and then this rate, and its relation to the inherent optical properties, and remove the information that is not of interest. So for example, here, if you want to know the concentration of CDOM, then from the entire remote sensing reflectance, we have to remove the information of the CDOM and NAP and seawater molecules, so we know exactly how much is available. And this likewise can be done for CDOM as well as NAP. So one way to do this is to look at or develop algorithms So once we develop algorithms, it gives us a framework as to understand the variations of these concentrations of different optically active constituents in time as well as in space. If in space. So I'll show you this one of the papers that we published for the chlorophyll A. So one of the problems in the remote sensing is that the concentration of chlorophyll A are severely overestimated in coastal regions because of the bio-optical complexity. So we used the same relation, that is remote sensitive reflectance, the relationship between the remote sensitive reflectance and inherent optical properties to develop a 3-wavelength index. That is shown on your screens as lambda1, lambda2, and lambda3. So here the objective was to identify 3 wavelengths that can give us the accurate concentrations of chlorophyll A, where the lambda1, it is the primary peak or it's the secondary peak of chlorophyll A. Lambda2 is used where we remove the effect of the CDOM and the NAP from the overall remote sensing reflectance signal. And the third wavelength is used to remove the backscattering effects from the remote sensing reflectance. So what we get is remote sensing reflectance that is, or let's say in practically, it is closer to what is closer to the influence of chlorophyll A itself, and not has the influence of CDOM or NAP in it. So we termed this algorithm as Goa University Case2 or GUC2 in short, and published this as an index. So CMCHL, that is the modeled algorithm, which was validated. So we used a diversified set of data that comes from SATCORE, that is a repository from the Ministry of Earth Sciences, maintained by INCOIS in Hyderabad. And then we have data from NOMAD, that is the NASA dataset. So we compared the performance of GUC2 with very many indices that are mostly used by the ocean science community. So we saw that the GUC2 outperforms almost all of them in our case. So for this, we use the same sort of data set in testing all the algorithms. And then the figure 9 here is showing you the matchup. So this, because the 663 or the 623 in the previous slide, the second wavelength, that is 623 waveband, that was not available in any of the remote sensor but MARIS. So now it has been included in Sentinel series and in the Sentinel series. So because of this restriction, we could only use the MARIS, and then we applied the algorithm to the wavebands of MARIS, and then compared that with the in situ concentrations of chlorophyll A. And then it worked pretty well over a diversified data set. Now, speaking of the estuarine coastal waters, we understand how dynamic they are when we are looking at the physical processes. For example, there are many external forcing agents like tides or winds, and then there are internal processes like convective processes that may induce the mix or and flux of the matter. So once there's a transfer of matter, means that the optical signature of that water column will change. So we wanted to test that this change, or if the algorithm can predict these changes in the estuarine coastal waters. So we chose a larger region, which is the Chesapeake Bay, and then we used many in situ data sets. And then we chose a time of September 2011 to January 2012. So in September 2011, there was a cyclone, Ike, if I remember. And then there was a huge freshwater discharge in the Chesapeake Bay, as you can see in panel B, that is showing the river discharge. And then it calmed down. So we wanted to see if all these surges can be picked up by the algorithm itself. And we applied this using the algorithm and tested the modeled chlorophyll algorithm, chlorophyll concentrations with the in situ chlorophyll concentrations. And it did well for these harsh conditions. And also, this was applied to the MERIS sensor. So for a total number of data points of 455, it worked very well in these environmental conditions. So what it all means is that we have an algorithm that we can apply to different environmental conditions, and it still gives us accurate chlorophyll concentrations in comparison to what is available in ocean science community. So it can be used in a variety of different forms. So next, what we did is we also developed this similar sort of index for the total suspended matter. And here we did what novel approach we adopted was to include the different groups of bio-optical significance and then use that information in developing the algorithm. So here also we developed a 3 wavelength index with the first wavelength being 679, and that is 681 in the remote sensors, 679 from the in situ measurement, 695 from the 695 as the second wavelength, and 704 that is the third wavelength. Now here the 679 that is covered by the 681 nanometer waveband in most of the ocean sensors, and the wavelength 3 that is 704 that is covered by the 709 nanometer waveband in most of the ocean sensors. But 695 that we reduced after the partial derivative analysis and then several other testing. That was and then 695 was the second wavelength. We were confident about it, but 695 is not available in any of the sensors. So we could not apply it directly to the remote sensor and get a synoptic synoptic analysis like we did for the chlorophyll algorithm or GOC2. But regardless, we published this only with the in situ analysis. And then the algorithm is being shown as equation 5 in this slide. Now, so recently we have developed the algorithm for the CDOM itself. And then it is currently under review. And then we'll know about it. So here we use a similar approach where here we are trying to understand, we develop the algorithm and then we are trying to understand the plume in the area of Goa. So here I'll show you the 2 estuaries, if you can make out in this, as Zuari and Mandavi estuaries, and some other parts of Goa region and the coastal part. So this is from the OLSI data that is on board Sentinel-3. So it gives us somewhat good resolution in the small estuaries that are shallow and then are optically complex. And then we do this analysis with the perspective of the freshwater discharge. As we know, the region is severely influenced by the Indian summer monsoon caused river discharge. So there is a lots of amount of terrestrial discharge that is coming into the rivers and then being transported into the coastal seas. So that was the basic fundamental reason why we chose to develop the algorithm and understand the application in terms of ecological monitoring of pollutants. And then this, we use the coastal data set that is also turbid and then case2 type to understand that how the light or how the underwater light field is varying over the large coastal region, and we studied the radiant heating rates for the entire western coastal region. So here, if you see that we have different OWT1, OWT2, which are the optical water types. So it means that these are different groupings of the remote sensing reflectance spectrum. So optical type 1, that is more offshore, they are clear waters, and then optical type 4, they are more near to the coast and are more turbid. So what we see is that the radiant heating rates in the table, if you see RHR, that is radiant heating rate, given in units of degree centigrade per hour. And then in the optical type 4, the heating is highest among all because the absorption was more. So now we have to consider that the euphotic depth of the clear ocean waters is much higher compared to the euphotic depths of the coastal turbid waters because the light is either absorbed or scattered very close to the surface. and in the open ocean waters, it travels farther down. So through this study, we came to know that the amount of, if we integrate the entire radiant heating rate, the amount of energy in the coastal deeper regions uh, is somewhat equivalent to what is being stored in the shallow turbid waters. So what it means that because there is more bio-optical complexity in the coastal water, in the turbid waters, in the estuarine coastal waters, turbid waters, they can capture or they can retain more heat that is comparable to the heat that is stored by the open ocean. So this was a significant result from this study. Now, the studies that I've shown you are more of purely bio-optical in nature. Here, we also wanted to test how the stratification changes this bio-optical complexity. In the region of Goa, this is Zuari estuary of Goa. So stratification is determined by the Indian summer monsoon related freshwater discharge. So during the monsoon season, we have a strong stratification in the region that decays in the post monsoon season, which is from October to January, and then in the pre-ISM season, pre-monsoon season, that is from February to May, it is completely extinguished. So it is completely controlled by tides in the pre-monsoon season. So here we determine the state of stratification using the in situ data set with the help of the CTD instrument. So this was done for a number of years, and then we used SATCO data that was the program of the Ministry of Earth Sciences from 2008 to 2015, 2017. So here we show that in the ISM, in terms of buoyancy frequency, that shows the strength of the stratification. In ISM, we have a strongly stratified state. And then in the post-ISM, because in the post-ISM season, because the freshwater declines and we have more tidal action, so the stratification or the maximum stratification that moves upstream in the post-ISM. And then the pre-ISM season, there is still some freshwater discharge, but there is more tidal action. So the maximum stratification site, it travels more upstream into the river. So using this information, we related the inherent properties at each station. So we saw that with respect to the chlorophyll concentrations, they were always present immediate downstream of the maximum stratification. And then this observation was stratification dependent and it varied seasonally. The CDOM values represented by the absorption of the CDOM at 440 nanometers, the minimum values were present where maximum stratification was there. And contrasting result was that the least ACDOM or the least CDOM concentrations were observed in the monsoon season, where the river discharge was maximum. So it means that the volume of freshwater discharge exceeds the volume of the estuary. So everything goes to the coastal seas rather than being retained in the estuary. And then the TSM concentrations were maximum at the site of the maximum stratification. And then as the stratification site shifted upstream, the TSM concentrations also shifted upstream. So this gives a brief overview of the entire measurements as the ISM and post-ISM scenario and the pre-ISM scenario. So now we have looked at the inherent properties and the stratification, that is hydrography. So with respect to this, we observe 4 different analytical groups. That is optical group 1, that is the river stations that are upstream of maximum stratification. Then optical group 2, that is maximum stratification identified from the n-squared values. Optical group 3, that is the maximum chlorophyll concentrations immediate downstream of group 2, that is maximum stratification. And then group 4, these are the estuarine stations downstream of group 3. So we looked at the spectral total absorption and the total backscattering by the particulates, and they were all distinct. And then we looked the contribution of each bio-optical constituent or the optically active constituents to the total absorption as in absorption budget, which is group 1, group 2, group 3, and group 4. So we can see that how much of the spectral absorption is being influenced by what optically active constituents. And then we did a little bit of modeling here by looking at the diffuse attenuation with respect to the total absorption and backscattering. And then for the 4 different regions of the estuary, we came up with 4 different spectra of remote sensing reflectance that can be used to track the water masses in the region of having different optical signature. So for example, group 1, which is panel A, that will represent the riverine stations. Group B will be the maximum stratification where the maximum TSM concentrations are observed. And then immediate downstream of maximum TSM concentrations is the maximum chlorophyll A, represented by a different remote sensing reflectance spectra. And then the usual estuary, represented by the panel D. So these are all different ways on how to generate a better or how to better understand bio-optical complexity in coastal and estuarine waters in relation to the open ocean waters that are pretty clean. I mean, I'm not saying that there are no problems in the open ocean, but phase 2 waters are much more troublesome because they impact our human lives directly. So coming to the end of this presentation, there are certain issues that I want to speak about. That in situ data is very important for these kind of studies or for any study. And then collecting in situ data and then training the manpower and then going on the field, collecting data, that is actually very, so we have to have better techniques to do it in terms of instrumentation. And then the performance and accuracy of the algorithms, they need to be constantly improved. There have to be more engagement of the community in terms of the application of these algorithms. And then from the perspective of the remote sensing sensors, the resolution has to be improved for the shallow estuaries and coastal regions. So there's a large-scale regional in situ monitoring programs required under one centralized database with public access, so that the application-wise this can be boosted, so that we can know more about this on public forums. And then the influence of the atmospheric conditions on the remote sensor reflectance itself in form of atmospheric correction, it requires a better understanding in Indian context. And this I'm saying only for the coastal and estuarine waters. So in conclusion, that we can say that instead of focusing on different regional algorithms for optically complex waters or case 2 waters, optical diversity should be considered. This would help in understanding algorithms and uncertainties at large while being applicable over larger regions. The use of algorithms for satellite monitoring of regions are just as good as their statistical significance. So without improvement, they will simply be out of use or of no use. So they have to be constantly tested with new data set and then improved upon. So there are many algorithms available in literature, many, but they have to be fine-tuned. So that work has to be taken to fine-tuning them for different larger regions so that more application can be done. And then algorithm improvement and has to include the environmental conditions such as dynamics, estuarine processes. All those things have to be taken into account as well when we are developing algorithms or doing studies like that. So many scholars, students, PhDs have worked in the remote sensing laboratory here, that they have collected data and maintained it, post-processed it, pre-processed it, and then have done the satellite analysis for many years. So they have to be thanked for all of this work. And the data collection and laboratory has been running at Goa University with the help of grants from the Ministry of Earth Sciences, Indian Space Research Organization, the Naval Research Board, and the Department of Science and Technology. So we are grateful to them as for the grants. Thank you. I'll take questions if you have any.
Thank you, sir. Any questions? You can unmute and ask the questions.
Hello.
Sir. Go ahead, sir.
Good evening.
Arjun
Sir,
sir,
this
you are
Srikanta speaking.
muted.
No, I have unmuted.
Sir, not you, Arjun sir. Yes, sir. Sir,
Okay.
please ask the question.
Sir, good evening, sir.
Good evening.
The Srikanta, on a personal visit to Dubai, but anyway, I had the opportunity to log into this lecture. It was good actually. My only question is, when you determine the reflectance ratio and things like that, make studies. There are a lot of turbulence and the liquid, the ocean water is continuously in motion, right?
Yes.
And they're continuously in motion. Then the impurities, the composition keeps changing, and it's got always there may be turbulence, and then things like that. It's totally dynamic, not static at all. Even the concentration of various things also will change, right?
Yes.
Yeah. So how do you take all these things into account when arriving at those figures?
Okay. So when we take the measurements, so we take
Hmm.
optical measurements, we take CTD measurements, that is telling us the salinity and the temperature values of the water column. And then we also look at the particle size distribution. This is only for estuarine coastal waters. So, what we are so actually in the terms of application, so there has to be a framework in terms of a module where we can test all the algorithms with the given set of hydrography or hydrodynamic conditions, and then reverse test the remote sensing reflectance that is being retrieved from the sensor. Yes,
Those things are taken into account while arriving. While
yes,
framing the equation and all that, right?
yes,
What you
yes,
showed some. Yeah.
yes.
Yes. Thank
So
you. Thank you.
all that data has to be checked.
Yeah. I understand now. Yes. Thank you, sir. Thank you.
No problem.
Thank you, Supriya, for giving this opportunity.
Thank you, sir.
Thank you.
Any more questions?
Hello.
Sir, go ahead.
Yeah, my name is Venkat Rao. I was the project director for OceanSat 2, and I just heard the lecture. It was excellent. I have only one just clarification that Adhikari has referred the OCM, ocean color data from Sentinel and MODIS. I was just wondering whether ocean color data from OCM 2 and OCM 3, latest OceanSat 3, has it been used, or are there any issues related to the data?
So OCM data has been used previously for deriving the salinity values. And yes, sir. And the sedum concentrations, that has been done.
How about OceanSat 3? We have more number of bands, 13 bands, I think.
Yes.
That also, are you using your research work?
Yes, sir. So we are using that.
Acha. Because you didn't just mention OceanSat 3.
Yes, sir.
I was wondering just why only Sentinel and MODIS when we have got our own data.
Yes, sir.
Okay, good. If you are using, I am happy. That's all.
Yes, actually, so for OCM3, we are testing it for modeling purposes.
Acha. Okay.
Yes.
But for our PFZ, we are regularly using OceanSat, no?
Yes, sir.
Yes, sir. Okay. Thank you.
Thank you.
Okay. Any more questions? Okay. Sir, I think no more questions.
Okay.
We can wind up, I think. If any queries, you can text me or you can mail us so that we can communicate. Okay. Thank you, Arjun sir, for your insight. Have enriched our understanding. And thank you for enlightening presentation. Thank you one and all for joining us.